Thank you very much for joining us today for the ICM webinar. The title of today's webinar is called Structure-Based Lead Discovery Using ICM Virtual Ligand Screening. So we'll be covering how to, the basics behind the method that we use. We will look at an example, screening to a cyclooxygenase receptor, where we can screen. I'll show you how to set up the receptor and how to screen a database of compounds to that receptor, and then how to score them and how to interact with the results. So my name is Andrew Ori. I'm a senior scientist here at MolSoft. And if you have any questions after the webinar, please feel free to email me at this email address or give me a call. Other ways to keep track of new developments in ICM is to follow us by Twitter or Facebook, or click on the news link, and there's a link there to show how to join our mailing list. Great. So thank you. So we'll begin. Right. So the virtual ligand screening is basically you can dock and score millions of compounds to a structure, protein structure, or to a ligand-based way, which would involve using a 3D pharmacophore model, which is we use atomic property fields. But in this webinar, we will just concentrate on structure-based virtual ligand screening, and you can screen to a single receptor, or if you have multiple crystal structures, you can, or multiple models, decent models, you can screen to an ensemble of those to incorporate induced fit or flexibility in the receptor. We also have ways, another way to incorporate sort of reasonable estimation of induced fit would be to have a hybrid map and allow explicit atoms to be flexible. For example, you could have a map of the receptor and would allow, for example, each hydroxyls of serines, threonines, or to be flexible in those maps. So there's a way to do that. We also have a way to perform covalent bond screening, which is slightly different than the regular screening. And we also have ways to screen focus libraries using Marcouche generated libraries. Okay, so we just start with some basics of the docking. So for virtual screening, there's two components. You have the docking component, where you dock the ligand to the receptor, and then you have to score that pose, and that will give you a score, and that enables you to rank millions of compounds to that receptor. So basically, docking is the modeling of the binding of macromolecules between themselves. There's docking would be you have protein-brain docking or small molecule docking. In this webinar, we're talking about small molecule docking. So factors that determine specific binding. You have the early docking methods were based on shape complementarity, lock and key, basically. So it's like a geometry fit method. And also you have the property component. So ideally, in your receptor, you're matching hydrophobic atoms with hydrophobic atoms, hydrogen bond donors with hydrogen bond acceptors, and then you have the electrostatics, so positively charged to negatively charged. And what you're looking for is to identify a stable complex where the bound conformation represents the global free energy minimum. And you achieve this by maximizing favorable and minimizing unfavorable interactions. And at the bottom, there's a paper here by Ruben Abagai and Max Totroff who developed the MolSoft's ICM docking method I can make available after the webinar for you. So docking as a global energy optimization problem has two components really. It has an energy, you have to have a decent energy function, and you also have to have a way of an efficient search algorithm. So the energy function needs to be a fast approximation because you're going to be screening maybe millions of compounds. It needs to be reasonably accurate, and ideally the global minimum should correspond to the near-native conformation. So different methods have different ways of different energy functions. So you have molecular mechanics force fields, and also you have where physical terms and parameters are based on force fields such as ECEP or Merck molecular force field. In ICM we use ECEP for the protein and Merck molecular force field for the ligand. Or you have ways of developing the energy function using statistical methods, such as the potential mean force method, which uses the statistics of interatomic distances that are observed in protein ligand complexes. So the search algorithm on the right hand side, ideally you need to quickly locate the energy minimum over a very rugged energy landscape. So there are geometry-based methods using rigid body followed, so the ligand and receptor are rigid, and the small local minimization. There are other incremental construction methods that are used, or genetic algorithms. Sorry. But we use, in ICM, we use a Monte Carlo method called biased probability Monte Carlo. So this method is described by Ruben Aragon and Max Tostroff in JMB in 1994. I can provide that for you. But basically, the Monte Carlo minimization involves random steps. It's a stochastic method, so you're randomly sampling torsions of the ligand or positions and orientations of the ligand. So you randomly sample, and it's followed by a local gradient minimization step. And then you compare the new energy of the ligand compared to the previous one that you had. And if it's improved, you accept the conformation. And then there's some statistical approach to determine whether to keep that conformation or reject it. And then you go back to step one and repeat, depending on the length of your simulation. So by default, we use grids for the receptor. But as I mentioned, there are ways to incorporate a grid plus explicit atoms if you have flexibility. Or you can use multiple grids, which is a method called 4D docking, to incorporate receptor conformation flexibility. So you'd have multiple grids for your receptor. So the ligand position and internal torsions of your ligand are optimized by the stochastic Monte Carlo search method in internal coordinate mechanics, which is ICM, which is our basic framework. There's a local gradient minimization after each random move. The ligand is continuously flexible, and the receptor is represented by pre-calculated grids. So the energy terms we have are van der Waals, steric repulsions, and attraction, electrostatics, hydrogen bonding, and hydrophobic interaction. Inside the maps, we also have another van der Waals potential, which is cut off here, which allows some clashes, minor clashes between the ligand and the receptor to incorporate softness in the docking. Okay, so when you have your protein and you're docking to it, some issues that you need to maybe look at before docking or screening. So the first issue really is obviously the quality of the X-ray structure. Ideally, you'll use a high-resolution X-ray structure. It's important to identify where the crystallographer has not seen any density for the side chains of the receptor. So there may be missing residues, or there may be missing atoms, side chain atoms. If there are missing side chain atoms, when you convert from a PDB structure in ICM to an ICM object, ICM will add the missing side chain atoms to that. So it's important to look at those and check that the orientation is correct. If there are missing residues, you would obviously see a gap in the chain. So one way to fix that would be to maybe model that region, or if it's away from the pocket, you may not need to worry too much about it. High B factors may indicate that there is a considerable flexibility in that region, or so you may need to be careful there. And if there are clashes in the structure, you may need to optimize or minimize the binding pocket. Some special features that you may want to consider is your ligand a covalent ligand? If it is, you need to make some adjustments to the map to incorporate the covalent bond. There also may be, you may have to change the charges of the protonated or you may need to protonate a residue or deprotonate them. And also, are there any ions or in the pocket that enable the ligand to bind, the pose that it should show? Or, for example, there may be a water molecule that might be important as well, which you may need to keep during the docking. You may want to consider the druggability of your pocket. Ideally, your pocket will be hydrophobic, reasonably buried. If it's a protein-protein interaction site, it will be very shallow pocket, most likely, and exposed to the solvent. So you may want to be careful exactly where you're screening to. And to identify the pocket, we have an algorithm called ICM Pocket Finder, which will identify the pocket for you. So receptive preparation. As I mentioned, you need to convert the PDB file to an ICM object, whereby hydrogens are added and missing heavy atoms are added as well. The polar hydrogens are optimized, and atom types and partial charges are assigned. Okay. So some special cases you may have, as I mentioned earlier, you may need to undertake homology modeling in the pocket. You may need to sample alternative side chain conformations. You may need to regularize the structure, but if you've got a high resolution structure, these may not be necessary. Okay, and then once you've docked your ligand, we have a scoring function, and the components of MolSoft's scoring function are the internal force field energy of the ligand, the conformational entropy loss of the ligand from outside the pocket compared to inside the pocket, the receptor ligand hydrogen bonding interaction, the solvation electrostatic energy change, and hydrogen bond donor acceptor desolvation, and hydrophobic energy. And each of these components have a particular weight, which have been optimized by MolSoft. But if you have this particular target, you can actually change the weighting factors for these. Okay, if you have any questions during the webinar, please feel free to use the chat, and I will try to answer them as we go through it, or I can answer the questions at the end. Okay, so just finally on ICM performance, the most recent docking and scoring evaluation using ICM was published in 2012, and this was the result of a industry-wide competition that was held by OpenEye, and the results were reported at the ATS meeting in 2011, I believe. And MolSoft did extremely well in this competition, and the results of this are shown in this paper. For some success stories, you can go to our website, and they are listed here. So, for example, just some examples. Here we had 1.2 million compounds were screened to this target, and 241 compounds were tested experimentally, and a number of hits were identified. This one, for example, 169 compounds were Cambridge database, which is about 500,000 compounds were screened, 169 compounds were tested, and 16 compounds had activity. Thyroid receptor, 250,000 compounds were screened, 75 were tested experimentally, and 14 antagonists were discovered. So these are listed here. Okay, so practically in ICM, if you have, in order to do virtual ligand screening, you need the ICM Probe plus virtual ligand screening package. And so when you open up the graphical user interface, it looks like this. So the structure we're going to look at is a cyclooxygenase receptor, and the ligands for this receptor are pain drugs that can reduce pain. So the PDB structure is 4-COX, and these are the receptor for Vioxx and Celebrex and inhibitors such as indomethacin. So here we have 4-COX, which we have 4 different molecules here. We're going to look at the A molecule, the A subunit. So what we would do is just delete, and inside the A subunit is an inhibitor called indomethacin. We can just zoom onto that. So we're going to screen a small database to this pocket. So just for clarity, what we do is delete some of the other molecules, for example, the other subunits. So we're just left with the receptor, which is the A subunit, and the ligand. So the most important thing when you're setting up the project is to make sure that the ligand is out of the pocket. So here we have one object with 2 molecules inside. So we need to move the ligand out. If we don't move it out, then the ligand will be incorporated in the maps, and so there won't be any space there to dock to. So we need to move it out of the receptor. We right-click and we choose the option to move from receptor, move from object. Okay, so now we have two different objects. There's a question regarding hydrogen atoms, which I'll address probably at the end. I think that might be the best approach for that question. Thank you. So here we have two objects. So we need to convert. Now we need to convert this the protein receptor to an ICM object. The way to do that is to right-click and say Convert PDB. And so you need to, if you've added hydrogens, or this process will add hydrogen. So we need to optimize the ideal hydrogen bonding network in the pocket. We also need to optimize like asparaginins and glutamines where the orientation of the side chain is unclear from the density. And the same with histidines, we will optimize the hydrogen bonding for those. So you select those parameters. And depending on the size, this may take a few minutes to run the conversion. So I have a already pre-converted version. Yeah, so now we can see that the receptor is an ICM object. It's got ICM written here, and we will have in the pocket we'll have hydrogen added to the ligand. So this is a reasonable structure. There's not a great deal of ambiguity in the pocket, but you may want to before screening, you would want to go through each residue and inspect them. You may also want to look at the electron density from the Uppsala site. So you can do that by going File and then Load File, Load Electron Density Map, and then you enter the PDB code and you go OK. Might take a while to read. So the map is here. So we can just contour that map around the pocket by selecting the residues you want to contour. You go Tools, X-ray, Contour Electron Density Map, and we go OK. So we had a question about the hydrogens and the positions of the hydrogens, and they're obviously critical. So I'll just I've contoured the whole map. But anyway, you can we can look at the you can look at the individual contours for the residues surrounding your pocket and how well they look in the map. Yeah. Okay. So the hydrogens are obviously critical. If the hydrogen bonding network in your pocket is incorrect or the charges are wrong, the score is going to be disrupted. And we had another question regarding an ion in the pocket. If there is an ion in the pocket, you need to make sure that when you set up the receptor that the ion is listed in the selection. I can show that in a minute. So you would have two molecules here. You would have the receptor and then the Het atom below this in your object. Okay, so now to set up this receptor, we can do we need to identify the pocket. In this example, the pocket is kind of obvious because we have the ligand inside it. Or alternatively, if we don't know where the pocket is, or we want to screen to allosteric pockets, maybe you can use ICM Pocket Finder, and this runs. Yeah. So it's Tools, ICM Pocket Finder, 3D Predict, ICM Pocket Finder, and this generates a number of different pockets here and here. It's got sketch accents. So the ligand it identified accurately found our indomethacin pocket here with the green surface. Okay, so to set up the project, the most important thing is we need to tell ICM where to place all our docking project files. So we go to Tools, and change working directory. And then you can browse for the location where you want to place all the docking results and everything like that. So you're okay. Right. So I've set this to this directory. So now all my files will be placed there. When I go to docking, and we go to the second menu option, which is set up receptor. So we give the project name, and it's asking us for the receptor object, so 4COX. So this is our receptor object. This is the ICM command line language, which is A_atom selection, and then it's the name of the object, and then period. So if you did have a het atom you wanted to include in the pocket, you could put an asterisk here, and that would it would make everything in that object part of your selection. Now it's asking for the binding site residues. The pocket is this one, number 4. So what I did was run Pocket Finder, so this table is fully interactive. So I double-clicked on that, and it made the green crosses for the selection here. Yeah, so that's the place where we'll make the maps. We go choose. Basically, it's ready for docking now because everything's set up. One alternative way would be to select the ligand, because the ligand is in the right location. We can double-click on the ligand and then say define site around select a ligand, or you can use the Pocket Finder. Okay, so we just run OK, and you may not be able to see it, but in the middle of the pocket we have this sphere. It's just a probe. It's just asking you where you want to start the simulation. It places the probe where the ligand will start sampling the pocket. So the probe is placed by default into the middle of the pocket. You can move it using the middle mouse button, and so the only real reason why you may want to move it is if the pocket is particularly large and you want it to sample, to make sure it samples a particular region, or if there is a known interaction that you want to, you may want to move it closer to a particular residue. But by default, it's in the middle, which is a reasonable place, and it's a random sampling anyway, so it's going to try and sample as much of the pocket as possible. We go OK, and that brings up this purple box. Okay, so this is where we will generate the maps. You can change the size of the box by clicking and holding on the corners. If you have a known ligand in the pocket, you may want to display the pocket in CPK, and that will, and maybe the surrounding residues, and then you can make sure that the box fully covers the binding site. In most examples, that the box is usually by default pretty reasonable size, but you may want to shrink it a little, or make it larger if it doesn't encompass the whole region. Okay. Just for clarity for the webinars, I'll make it as small as I can, just because the making map step takes a minute or so to make. So you can see it's making grid maps for the van der Waals interaction with a carbon probe. This map is a G electrostatics interaction map. And we have other hydrogen bonding grid potential map, and hydrophobic. Okay. So the maps are now made. And then we may want to just double check what files ICM has produced. So if we go to the our directory here, we see that it's produced a number of different. The title of my docking project was webinar, so these are the files here. So we have the maps, we have a log file, we have a probe object, which was that probe that we used to determine the center of the pocket. We have our receptor object, and we also have a DTB file, which is a text table file. And we'll have a look at that file in a minute, because there's a number of different parameters that you may want to change in that file. Okay, so we're doing, in this webinar, we're doing virtual ligand screening, high throughput. So we need to, we're most likely going to be docking an SDF file, which will be a collection of mol files. And these can be millions of compounds in size. So we set up the receptor. We're not doing interactive docking, so we ignore this menu option. You can dock a chemical table if you wanted to using virtual screening, but for this example, we will use we'll set up batch from SDF file. So there are other options here. You can have a file, an ICM object file with multiple ligands inside. Or you could, if we have another product called MolCart, which is a chemical cartridge where you can store and manipulate large databases. Is also, if you have a very large SDF file, you need to index it. That will speed up the screening. In this example, we haven't got an index, but if you need to index your file, you would go to Docking, Tools, and Index mol file. So you just browse to your SDF file, and then give the index a name, and it will store it. So if you're screening, more than 1,000 compounds or something, you may want to index them. Yeah. So for this example, we're going here from SDF file, and go to Browse. And in this example, we're using an SDF file called Celebrex50, which is a database, a very small SDF file, just for speed in the webinar, which contains fragments of COX cyclooxygenase inhibitors. So we browse, we tell ICM if it's a mol file or a mol2. We want ICM to build hydrogens when it docks it. We also want ICM to assign charges, and it will also convert the 2D depiction of your ligand to 3D. So go OK. So I'm just going to show that SDF file so you can see exactly what we're docking. Okay. So if you have ICM, you have this file already included. So we have Celebrex, which is a cyclooxygenase inhibitor. We also have a number of different fragments here, 50 of them. So this is what we're docking. So before we do the screening, so basically we're here. We set up the ligand. We set up what we're going to screen. So before we start the screen, we need to double check some preferences. So we go to General, and you can choose to sample race mix centers, double bonds, cis trans. You can choose to keep the carboxyl neutral. And probably one of the key things here is the charge groups. You should really set it to auto, and that will automatically use our prediction of charge and charge the ligand according to that prediction. And you go OK. Then we go to docking again, and in preferences, because we're doing a virtual screening, there are some preferences that we can choose when we scan the database. So the first preference is the score threshold. This basically, when we look at the scoring, the lower the score, the more likely that the ligand is a binder compared to a non-binder. And a reasonable threshold is usually -32 for that. But if you want to check exactly what a good number for this for your particular project would be for this example would be to take indomethacin and then redock indomethacin and look at its score, and then that would give you a good threshold. So any compound that scores higher than this, ICM would discard, and we won't make an object for that 3D interaction. But if it scores lower, which is better, we will keep that object here. You can also add, if you have a million compounds, you may not be interested in the results of some of them because they may be not drug-like. So you can say the minimum ligand size is 100, for example. These are like the Pinsky's rule of 5, automatically included. But you may want to increase the ligand size or hydrogen bond donors and acceptors. Yeah, okay. So this is critical. So if for example you have a very open pocket, -32 may not be a reasonable score. You may want it maybe a bit higher, at like -20. So if you run screening, you won't get the 3D objects out if they don't score lower than -32. So it's important to check this. So once you're happy with the preferences, you can run this. You can start the screen. Oh yeah, there's one thing I wanted to say. But before the screen, you want to go back to that this file, the .dtb file, and in that file there are a number of different fields. Most of them are not that important to change. For example, you changed this one, for example, max hydrogen donors, you've changed in the preferences already. Just one which might be important is the log P. Sorry, print. I want to look at it. Yeah, so the maximum log P is 5. So any ligand that has a predicted log P higher than 5 will be taken away. So you may want, I think this is a default value, which is pretty reasonable PK, but you may want to increase it slightly. And obviously it's a prediction of log P. There are other fields that you can have a look through this file and check them. This one, for example, is where you're binding to. It gives the ligand, the residue numbers that you're binding to. This is important. This is where your this will be automatically set up for you anyway. But when you do it in the GUI, it's the location of the SGF file that you're docking. This is the type of SGF file. So that there's two. You could have mol file and mol2 file. They're basically both of those types of files can be docked. Okay, so when we're happy, we could, with the setup, we've checked the pocket, we can start the docking running. So to do that, we go to run docking batch would be the default way. But if you are trying to split your docking up into individual batches, maybe to run on different nodes of a cluster, for example, you can do that using run database scan batch. And so it's asking you the name of the project, which is webinar, and you maybe want to start from compound 1 to compound 5000, and then run that screen 1 to 5000 on one node, and then you may want to go from 5001 to 10,000 on another node. So you can run multiple jobs this way. But by default in the GUI, you go to docking, run docking batch. It's asking for the project name, the output file, which has the name answers. You can change that to whatever you want. This will be the file that contains the 3D objects of your ligand. The docking thoroughness is 1 by default. You can increase that, but if you're screening millions of compounds, this is a pretty well optimized number. This is the length of the simulation. If you're doing 4D docking, you may want to increase it slightly, just to increase length of simulation. You can store a stack of confirmations for each ligand, so it docks and it gives you the score for the highest one, but then you can go through and see other energetically favorable poses. So if you want to score each pose for a single ligand, you can check here. The problem with doing that is that it will slow down the screening, because you're docking the ligand and then you're going through the stack of other energetically favorable confirmations for that ligand and scoring each one, so it takes a little bit longer to run. So we're using these default, and we go OK. And you can see at the bottom a progress bar to see how well how far the job is progressing. You could also go to Windows and look at background jobs, and you can see that the job has been running for 16 seconds. So it takes a little bit while to run, but while it's running, you can actually view the hit list. So we can go to docking and choose Make Hit List. So the multi-object file, we asked it to be called Answers. The name, so this is where all the answers are located. If you're doing it in the GUI session, the path is already there, so you don't need to browse. We want to process this results and look at the 3D structures. If we have, maybe you ran the same series of screens 3 times, so you can look at all 3 of those and then check the lowest, the best score from each of the different runs. You can go OK. This option is if you just want to select maybe the top 100 hits. If you're, for example, a very big hit list which doesn't display with the memory of your computer, you can just select the top hits, maybe the top 1,000 or so hits. But we'll just go OK and run. So you can see that we have a new table, new chemical table at the bottom. And in this table we have a column called score, and we can sort by the score. The screening hasn't finished, we haven't got all the results here. And then we have number of atoms in the ligand, number of flexible bonds, torsions. Then we have values for different energies in that for this docked ligand. So if you want to look at the confirmation of that ligand in the pocket, you can just do a single click and that display. So this is finished now, so the whole screen's there. So let's just clean this up a little bit, the display. So if you just, you can browse through the docked complexes by single click, or just using the cursor key. You can go through your hit list, displaying the hydrogen bonds. Yeah. We had a question about whether when you run the docking and then you stop it for some reason, can you restart it from the position? What you would do, because once this docking is run, we get a file called an output file .ou, and if you look at the bottom of this file, it's a text file. So in here has all the information. So it tells you what your setup parameters were. And at the bottom of, so if your docking crashed or whatever for some reason, you would see at the bottom the last docked ligands. So we had 50 ligands in our SDF file. The last one was 50. So if you wanted to rerun this, we could go to docking, scan batch, and then enter 51 here, and it would carry on and run from that point onwards. Yeah, so this is the hit list. You can it's fully interactive. So you can plot, for example, you can make a plot of score versus molecular weight, or score versus number of atoms. Here, it's fully, so the lower the score, the better. So you can select. This is fully interactive between the ligand, the plot, and the table. There are other ways to make a selection. So if you imagine we've done the screening and then we're trying to cherry pick some compounds for experimental testing. One way to do that would be you can use a fingerprint. So you want to select a diverse set, basically, of ligands to test from the hit list. So you can click on this button here, which is a clustering, and this will run a fingerprint clustering. And you can select centers from each of the clusters. So select centers by minimum score, and go OK. So you can see we have the blue is the selections. So from this table we have 50 records, and we've selected 19, and so we can just copy that selection to a new table that we could send to our collaborators for testing. Another way, which is probably a better way to cluster, is using atomic property fields. So we have this. We want to select a diverse set of compounds. So this will cluster by how well each ligand is superimposed in a pharmacophoric field. So we may want to select a diverse number of compounds for testing. So we can run this cluster by APF. So you have a similar thing. You have a new tree. So the compounds in the same branch have got similar pharmacophoric properties in that position in the pocket. So that's a useful way of looking at the results. Other ways, in the right-hand side, in this there's this button here gives an extra panel, so you can look at different properties. So you can add a column of charge. Maybe you know that your ligand may have a negative charge or something, or so you can click here, press charge, and you'll get a new column with the charge of each of these ligands. You may know that I don't know, maybe a charged nitrogen makes a key interaction with a specific residue, and you're just interested in those residues. So what you would do, you would select the residue that you're interested in, and then run this, and it will tell you whether any of these ligands make that conserved hydrogen bond. You could also have a distance restraint as well. So you want this ligand to be not in this side of the pocket, so you can have a distance restraint there. You can also look at cis-trans amide angle. Sometimes the amides might be in a cis conformation, and that's pretty energetically unfavorable. So you may want to take those, or you've got a lot of yeah, it's not an ideal geometry. So you may want to remove those that are in the cis conformation. But ideally, in a good pocket, you probably won't have those. You also have non-overlap with selection. That's another way of making sure the ligand is out of the pocket. There's also other visualization tools in this panel as well. So you can look at the pocket surface. It's just building the surface. Or you can look at the pocket skin. So the pocket surface shows the skin. So they're coloured by binding properties. So there's an option called Pretty, which displays the ligand in a slightly nicer, better representation, so you can just see the key residues in the pocket. You may want to look at the individual atom energies for each atom in your ligand. Run display CPK. So these are sort of an estimation of the energy that for this interaction. So the lower the energy, the better. I had a question. What is the MF score? That's a different. It's not ICM, MolSoft's ICM scoring. It's potential of mean force scoring method, which is a statistical scoring method, which is used by the generals. People that don't use ICM may more like to use that. So it's just another scoring method that we have. We also had another question. Can we give different weights for scoring parameters? You can, yes. You can also do it using this table, so you can weight particular contributions, and then you get your own score here to do that. Okay, so getting to the end of the webinar, I just wanted to show relatively quickly the 4D docking setup. So 4D is for multiple receptor conformations. It's to incorporate flexibility into your receptor. So it's basically the same approach. The only thing that's different is that we need to make sure we have all our conformations in a single object. So here we have 3 nuclear receptors. The first one is converted to an ICM object. We want to place, these are all the same receptor, but their side chains are slightly different in the pocket. So we want to dock to those different conformations. So to do that, we need to place all 3 of these into a single object. So we go to Mole Mechanics option, and we've got an option called Impose Conformation. And it's asking us which is the main object. The main object is the ICM object here, and the source objects are the other 2 objects which we're going to superimpose. So I'm just going to select the other 2 like that, and we go OK. And then this superimposes them and places them all into one file. So now we can see in our main object we have the stack. So if I display this, we can go through the different conformations for this receptor here, or you can play them. Nice movie. So to set up this, we place the ligand, It's exactly the same basically, except there's one different step. So we go to docking, set up receptor, set up the receptor, and then you go to docking, and you choose flexible receptor here, sorry, and set up 4D grids, and that will make grid maps for all three of our receptor conformations. And then you go docking, and exactly the same. You go to the batch from SDS file, and then run docking batch, and then you make a hit list. And in the hit list, you'll see an extra column. And finally, if you set up your docking project once and you may shut down your computer and you want to go back to it, you need to go to docking, open project, find the location of your project directory, which would be set from this change working directory option. Enter the name of the project and go OK. And so this displays the receptor. We can just go docking and remake the hit list here. And if you did 4D docking, this column is the most important one because it will return the receptor number confirmation that is most favorable for this ligand. So it's displayed there. Okay, so basically that's the end of the webinar. As I mentioned, you can email me here, or I'll be here for the next 10 minutes, 15 minutes. If you have any questions, please use the chat, or you could probably talk as well. If you just ask in chat, I can put your mic on. Yes, and the next webinar is next week, August the 26th, and in that webinar we'll be looking at ligand-based methods, so using atomic property fields, and that includes ligand-based screening. So thank you very much for your attention. Yeah, if you want to ask questions, please use the chat. Just had a question. When fitting fragments, pocket is not completely filled with the fragment. Does ICM fill the remainder of the pocket with water? That's a good question. We're actually quite advanced in a new method for fragment docking, which Max Totroff and Ruben have been developing, and that will be released very soon, which will take into account these things. By default, the docking, you would have to incorporate the waters that you know that are there into the screening in the receptor setup, so you can include the waters for fragments. But we've got some new fragment-based methods. That method will be released soon, I think. Yeah. So to answer the question, you'd have to fill the water manually with the pockets and include them in the maps with the docking. We had a question, does ICM take advantage of GPUs? Yeah, there is a way to do that. You can run it on, yeah. Also, what's the difference between surface and skin? The surface is the, if you're referring to the docking hit list, we display the pocket, the receptor pocket as like a surface, which has the properties, but the ligand pocket representation allows you to visualize more spaces, the sort of the spaces where you can maybe build into if you're designing a ligand based on that hit. I hope that helps. So by default, there are two different representations: the pocket surface and the ligand surface. The ligand surface it gives you a better visualization of where you can build out of that ligand. Yeah, we had a question about validating the method. So we can offer trial versions. Usually, it has to be approved by MolSoft, but we can offer 2-week trial versions. If you email me, I can send you instructions on how to do that. If you have a question you want to use the mic for, just let me know in the chat, and then I can open your mic up. Yeah, I'll be posting the webinar online so you can view it again. I'll send you an email with that link. So if there are no more questions, I can take the other questions offline, I think. So thank you very much for your attention, and thanks. And the next webinar is next week. Thank you.